Hi, good evening. Today we are going to see about DNA repair mechanism. In DNA repair mechanisms, we will be talking about nucleotide excision repair, where we will see how the UV light causes pyrimidine dimers and how that excision is important. We will talk about base excision repair and the enzyme that is involved in it. Also about mismatch repair, non-homologous end joining, as well as recombination double-stranded DNA repair. Let's see how nucleotide excision repair happens. First, the damage is recognized. These are the nucleotides. This can be either purine or pyrimidine. But due to damage, like the UV light, this is a pyrimidine and this is a pyrimidine. The pyrimidine form dimers, and this damage is recognized. After that, this area is excised completely. It is filled with DNA. Polymerase fills the gap. After that, the DNA ligase will help in ligating the ligating the nucleotides. Let's see more in detail about it. This is the healthy DNA. Fine. The bases are arranged complementary to each other in this way. Now, because of harmful UV light, this DNA, in this healthy DNA, this pyrimidine and this pyrimidine forms dimers in this way. So that a covalent bond is formed between this pyrimidine and that pyrimidine. So in this way, pyrimidine dimers are formed. This is the covalent bond between pyrimidines. Now this pyrimidine dimers are now excised by this nucleotide excision repair process. If this excision does not happen, then there are serious chances of having Xeroderma pigmentosa. It is mainly because of a rare genetic disorder where this excision doesn't happen properly, thereby resulting in this disease. Let's come on to this. Let's see the second repair mechanism, which is a base excision repair. We had already seen this is a base and this is the another base. These bases have to be excised if it is not properly aligned with each other or not complementary. Due to any defect, if it happens, these bases have to be excised. This is done by first identifying those abnormal bases. The DNA glycosylase is the enzyme which you need to remember in base excision repair. USMLE mainly concentrates on the DNA glycosylase enzyme. And after that, this base is removed by the glycosylase enzyme. That site is processed and that gap is filled by DNA polymerase and DNA ligase. If you want to know more about DNA polymerase and DNA ligase, you can refer to my previous video. What is a mismatch repair? Mismatch repair is mainly about it will correct the mismatched bases in newly synthesized DNA. So, in newly synthesized DNA, it will be unmethylated strand, and if there is any mismatched bases, then it will excise and it will resynthesize the correct sequence. This is what happens in mismatch repair. But what is that area of the DNA which is more prone for mismatches? Usually microsatellites are that area of the DNA which is more prone for mismatch, because what is a microsatellite? Microsatellite are nothing but there are tandem repeats. So when those repeats, in those repeats, there are high chances of mismatch. So this mismatch repair mainly happens due to microsatellite instability. What is that syndrome which will result in mismatch repair mutations? It is a Lynch syndrome. So what are the three important keywords that we need to remember in mismatch repair? Mismatch repair will result in microsatellite instability and it will result in Lynch syndrome. If you want to know more in detail about this, you can refer to my previous video. See, this is the mismatched basis. What are the non-homologous end joining? So first the DNA breaks, and that broken DNA, the ends are processed and it is undergoing ligation. How does this happen? Say for example, here the DNA is broken and now both the DNA strands are processed and it is been stuck together by my DNA ligase. If this doesn't happen properly, it will result in Ataxia Telangiectasia. That is mainly because of ATM gene. Let's see about recombinational double-stranded DNA repair. Here, the double-stranded, the DNA break is identified. Say here, the DNA is broken. This is the one is an abnormal template and the other is the normal template. The double-stranded break is recognized and the enzyme processes the broken ends to create strands. See, the double-stranded DNA is recognized. This enzyme processes the broken strands to create single-stranded overhangs. Once this single-strand overhang is created, so this is the damaged area and this is the normal one. With this normal one, a single-strand overhangs are created. With the help of mediators like BRCA1 and BRCA2, it will facilitate the strand invasion of the undamaged homologous DNA. Say with the help of BRCA1 and BRCA2, what will happen? This is how the nucleotides are added, so as to produce the new DNA. So whatever strand which was undamaged, this is the undamaged strand and this is the damaged one. The undamaged one will serve as a template for accurate repair through DNA synthesis. The repaired strands are also resolved, now restoring the original sequence. After that, the sequence has been restored. This is what happens in homologous recombinational mechanism. So when does this happen? This homologous DNA repair mechanism happens during the S and the G phases of the cell cycle, because only during this phase of the cell cycle, G2 phase of the cell cycle, because only during these two phases of the cell cycle, this sister chromatid is available. Only if this sister chromatid is available, then we can produce this homologous recombination mechanism. I have an announcement to make. I have an announcement for you. I am starting my mentoring lessons for medical graduates and also for USMLE aspirants. So if you like my videos and if you want me to coach you one-on-one or in a group class, feel free to message me in the comments and fill the Google form. Looking forward to meet you soon. Bye.